dari Royal Australian Chemical Institute mulai 2005 sampai sekarang. Beliau juga menerima Australian Postgraduate Award ya pada tahun 2006-2009, kemudian Commonwealth Scientific and Industrial Research Organization Postgraduate Scholarship, kemudian ARC Dekra pada tahun 2016 sampai 2018. Untuk mata kuliah yang beliau ampu adalah kimia medisinal, kemudian drug design and discovery, pharmaceutical analysis, dan pharmaceutical chemistry. Beliau memiliki research interest di bidang glycogen sintase kinase 3 beta inhibitor, a stimulator of steroid genesis ya. Kemudian juga di bidang pengembangan obat senyawa anti tubercular drugs ya untuk tuberkulosis HIV co-infection. Beliau memiliki banyak publikasi ya di bidang medicinal and biomolecular chemistry ya, kemudian organic chemistry, organic chemical synthesis, dan penyakit infeksius ya. Publikasi beliau sangat banyak sekali ya, tidak mungkin bisa dibacakan satu persatu, akan tetapi kita bisa menyimak ya hasil-hasil karya beliau ada di CV-nya di website dari Curtin University ya. Oke, baik. Tidak perlu berlama-lama, langsung saja kita simak perkuliahan ya. Kepada semua mahasiswa, kameranya standby ya, on cam ya. Oke, terima kasih Dr. Hendra, monggo.
Saya unmute sekarang. Mohon maaf, ini suara saya jelas tidak?
Oke, jelas dokter.
Jelas ya, makasih.
Jelas.
Ini saya juga di meeting chat, I just wrote something. Terima kasih untuk kata sambutnya dari Bu Yuni, memberitahu diri saya ke mahasiswa. Selamat pagi semuanya, salam kenal. Nama saya Hendra Gunarsoyo, asli orang Surabaya. Tahun lalu, awal tahun ini saya sempat ke Surabaya pas pulang, sempat ketemu dengan, sempat ke UNER, diajak ke kampus J, and yeah, no, it was fantastic. I did meet with Professor Sis, Prof. Sis Wanyono. Yeah, obviously, the biggest name in medicinal chemistry in Indonesia. Saya ingat persis ketika saya masih belajar medicinal chemistry di University of Sydney. Saya lihat di Indonesia apa sih kalau kimia obat textbooknya, lihat wah kimia medicinalnya Pak Siswan. Yeah. So just want to let you guys know, I'm Indonesian. I've been here for a while. And
oh iya anjir, laprak.
Ya, mahasiswa tolong di mute semua ya. Bu Valis, apakah bisa dibuat ini dari host? Albin Musyaf, tolong dimatikan ininya speakernya ya.
Terima kasih.
Mohon maaf.
Ini hari Sabtu juga. Ini saya mungkin ada sekitar 80 slide ya. So intention saya, look, my understanding ketika when I was speaking with Putri is that ini second semester undergraduate students in pharmacy. So my intention today is to actually give you give a bit of a introduction and background about the kind of biological and physiochemical barriers that drugs need to go through in order to reach the desired action and achieve what is intended. So the first sort of maybe half an hour, I will talk about the nature of the biological barriers. I'll talk about the nature of drug targets. I'll talk about receptors, proteins, enzymes, and so on. And in the meat, the real sort of flavor of the talk will be some medicinal chemistry alterations. So we always like to think ourselves as a person who has these appropriate tools that we can fix, we can break, we can make new bonds, we can change bonds, depending on what the environment that is the most suitable for that compound to bind to. So this is us. So it is our intention that students understanding or trying to learn pharmaceutical and organic synthesis to be able to equip themselves with these tools that will allow them to actually understand what makes a good drug and how can we further improve the qualities of those drugs. We are
mohon maaf Pak Hendra, mohon maaf.
ya,
Di kami suaranya agak ini ya, double.
oh no.
Iya, kurang jelas, mohon maaf. Dan slide-nya ini memang belum jalan ya. Slide-nya memang belum jalan. Oh ya. Ya, kami agak ini double.
Agak double ya. Kalau sekarang
sudah lebih jelas. Sudah jelas.
Mungkin saya ngomong lebih dekat ya, maaf. Ya.
Ya, terima kasih.
Okay, jelas. Terima kasih. Thank you for letting me know, Bu. So, for the start, in medicinal chemistry, so this is drug discovery, although it's taken from more than 20 years ago, the basic premise of drug discovery is more or less the same. We may come from different starting
This meeting is
points,
being recorded.
effectively, in the preclinical studies that is on the top corner here, there must be some discussion of what do we want to cure, right? So some sort of research team, they get formed, form the objectives, and they must start from starting material, right? Some sort of starting drug. Either this one is obtained from hydrophilic screening, or this is obtained from natural products, or this is obtained from repurposing of certain chemical for either similar conditions that targets similar drug targets. Either way, most of these novel chemicals that are synthesized or procured, the team will then need to get some sort of proof of concept that the drug, the compound, that is meant to be either an activator or an inhibitor or a modulator of certain proteins, when they actually administer that to some sort of animal models as well as in vitro systems, that they are able to actually see a function that they would like to see. And so this is the validation process. So this is forming that initial proof of concept that yes, this drug that is meant to inhibit certain enzyme X is actually useful in alleviating the symptoms that is shown by the animal model set. And so this, a lot of the academic drug discovery is actually based on this sort of area. And then typically the jump from the academic drug discovery to the sort of more industrial application is this barrier of scale-up synthesis, obviously. So you initially in academic labs, you probably do up to perhaps 5 to 10 grams. That's possibly maximum that an academic labs can do. But obviously, before we can go to humans, especially in phase 1, then we need to look at a lot higher. So typically from this academic process into the industry, there needs to be some sort of agreement with the industry partners. So for example, one of my mentors, not my previous mentor in University of Sydney, they actually just signed an agreement with Boeing Ingelheim that is worth almost 200 million Aussie dollars. So that is the kind of investment that is actually needed to sort of break through from the academic drug discovery to the industrial ones. And basically from there, look, I'm not going to talk too much about this, but effectively the typical fourth phase of the clinical studies. So in phase 1, generally the intention is to define what is the maximum tolerated dose that healthy patients can take so that they don't show any undesirable toxicity. Right? That's typically what the phase 1 studies is. Obviously, in a cancer setting, if you are doing an anti-cancer drugs, there is no point for us to actually keep anti-cancer drugs to phase 1 humans. So in cancer, typically they go straight to phase 2. And phase 2 and phase 3 largely similar. So this is the recruitment of patients that suit the disease category. They tend to sort of differentiate them based on how advanced the stages of cancers are. Phase 2 studies typically in a small number, and phase 3 tends to be in hundreds or thousands of patients. And basically, at this point, this is when the make or break actually happens. And typically, the lifeline of that drug is going to be known around this point. So, for example, you all would have heard about Ozempic and the small molecule version of Ozempic. So there are two now that's in the market. But last year, when I was following this up, that drug that was in Europe still is still they're at this stage they're negotiating with the Asian partner, with the Australian partner. Hey, can we advertise and can we put these drugs into your market? Right? So typically, if you like the money-making business, starts from about that point onwards. And then from there, typically, you have about 20 years to maintain your copyright. We all see what happens with Lipitor. Lipitor make a lot of money in the early 2000s. And then after that, obviously, the generic drug version makes it to the market. And obviously, newer drugs are actually. Right? So so crucial to that process that I just talked about is the role of the synthetic medicinal chemist. Mm. So the role of the synthetic medicinal chemist is exactly in orchestrating that whole concept of can we start with finding what is wrong about the disease that you're looking at? Can we pinpoint what is the abnormalities, what is the malignant signaling that happens with certain biological pathways, and then can we find a certain drug, a certain molecule that would allow us to actually study that disease better, and with the hope of eventually you can find a good modulator. Right? So when you combine organic chemistry, pharmacology, and biochemistry, which is obviously very relevant to the pharmacy students these days, effectively, the organic chemistry set allows you to have that, if you like, organic eyes, right, to look at the structure. It enables you to assess how drug-like your structures are. It enables you to assess how metabolically labile your compounds are, and also sort of telling you what are the important functional groups that would be useful for the binding of your compounds. The pharmacology in biochemistry is obviously handy when you are interpreting your biological assay, when you are choosing which in vitro model, which in vivo model you should be looking at. So really, these days, when we train medicinal chemists, it is no longer enough for students to just have synthetic skills, but also they need to have at least a basic understanding of how the assay is actually done. Right? So for medicinal chemists, structure-activity relationship is our bread and butter. This is effectively 80% of the time that my PhD students spend, and every medicinal chemistry PhD students spend, but also structured property relationships. And there is more and more, obviously, in silico or even AI that would actually allow this. Now, can I ask this audience first, if that's okay? Is everyone familiar with the concept of Lipinski's rule of five? Lipinski's rule of five. Would you like me to go through this again, just to refresh your memories? Or if anyone can please chat on the text box, that will be nice. So I'm not just talking all the time, so I can get a bit of feedback from you guys as well. Please review. Thank you, Natalie. So Lipinski's rule of five, as it suggests, it is a set of rules. Although Chris Lipinski himself actually said it is more like a guideline rather than a rule. There are four, so maybe I'll type it on the text box as well. Linsky's one molecular weight of less than 100 to H less than 5. Oh, sorry, not bad. OH plus and H less than 5. OH 4 plus 10. And finally, C log 3 less than. All right. So I just typed in the Lipinski's rule of 5. Just I'll delete the second one. All right. So if the students please look at the chat box, those are Lipinski's rule of 5. So the idea is that if a compound is small enough, so first one molecular weight is less than 500, and if the compound does not have too many hydrogen bond donors, so doesn't too many OH and NH groups, typically less than 5, and also not very polar, so the number of oxygen and hydrogen atom is less than 10, and finally, it's not very lipophilic. So the C log P is a measure of how lipid soluble your compounds are. Generally, if your compound is too lipid soluble, then you end up getting trapped in your in the matrix, right? In the protein matrix. So it tends to spend more time getting stuck in there rather than actually circulating around the body. So Lipinski is basically make these rules and saying that if the compounds satisfies all these 4 criteria, chances are your compound is going to be bioavailable. So the rule is, let me make sure, if compound answers all 4 criteria, compound is likely to be bioavailable. That's the So, so very quickly then, we start being able to see, all right, so to be a good drug, generally this is the criteria that we need. And then from the organic chemistry, from our high school chemistry understanding, doing organic chemistry, when we start learning functional groups, we then start to see if you like, the connection, right? So these are the set of the functional groups that we know, and these are the set of the criteria that we need to fill out. So when we are designing compounds, when we are implementing changes to the structures, we need to keep those in mind. Okay, so I hope that part is okay. Now, for this one, my intention is, I'll make this quite quick. The intention is to say that a lot of the drugs that we have these days are either natural products, directly derived from natural products, or they're inspired by the natural products. So anything from penicillin, the caffeine that we have every morning, to the painkiller, obviously morphine, and also this digoxin for health, for heart disease. So all of these guys, they all have alkaloids, so they're nitrogen containing. So you see this nitrogen here on the lactams, a lot of nitrogens, obviously the purines for caffeine, and as well as the morphine. Now, you probably, can I please check, you probably would have done this in your first year chemistry lab. So this is typically, perhaps not exactly a Seneca, but when your lecturers, when your senior lecturers, and when your professors gave you a mixture of acidic, basic, and neutral mixture, what are we going to do to be able to separate the 3 components, right? So, for example, in this case, if we know that this agatrin, which is obviously basic thanks to this nitrogen group, is somewhat embedded inside this plant, and we do want to take this out. So effectively, firstly, we need to make sure that we do a acid-base wash. So first wash, in this case, is basic. So on the organic phase, it will extract all the neutrals, including the ephedrine, whereas on the basic phase, obviously, that will decarboxylate, that will deprotonate your carboxylates as well as other debris. So if you like the basic wash, take away all of your acidic functionalities. Right. Following that, what do you do? Well, from the organic phase, you can then put HCl, and once you do HCl, then it's effectively you are reprotonating that nitrogen, obviously the avidrin, and that will go to the aqueous phase. Right? So at this stage, if you like on this bit, you are taking out all the acidics. On the organic phase here, for step 2, you're taking out the neutrals, and here you're taking out the basics. And so continuation, that was the same. And once you get the basics, and if this is really what you want, then you can further do an acid-base wash. So you effectively deprotonate again, your HCl salt, and from there it is a purification. Is my screen? Can everyone still see my screen okay? Yeah, okay. Now, for the next one. So the next one, again, I don't want to use too much. The intention is that when you get compounds from natural products, if you can get it from a reasonably big quantities, you can actually do that as a initial starting material to further some modification. So the jargon that we use for this is what we call semi synthesis. So what the famous example is what we with Taxol, which is an anti-cancer drug. So Taxol is double drug here, is actually very complex. But on the top of each trees, we can actually isolate, if you like, compound that is 5 or 6 steps away from salt. So the idea is similar. So if you wanted to make aspirin, you obviously need to isolate the salicylic acid, and salicylic acid is present in high amount in salicin. Likewise, if you want to make it if we want to make a more derivatives, then again, from feeding that you can get from the seeds, you can actually get already the right function in the right place. And so you just need to do relatively fewer segmentation rather than that. So this approach has been getting more popular. Obviously, it saves resources. The idea is to try to upcycle value of that product that we can easily obtain from that. This is, I think, this is my sort of my last slide on this part. Okay, so colleagues, there seems to be an
Yeah,
mistake.
Dr. Hendra, I'm so
Yeah.
sorry, but it seems to me that there is audio interference. We cannot clearly hear your voice.
Um, let me. Last week, I did something about this.
Yeah, it's still not clear, doctor.
Still not clear.
Yeah.
Let me. Thank you for letting me know. This, is effective, or am I sounding like a trouble now? No. How about now? Maybe police.
Yeah, it's better. Ya,
Um,
Bu Yuni jelas ya, Bu Yuni.
Iya, agak gemes.
Agak, nggih.
I think yeah, many interference in the voice.
Does this happen begini? Maybe I'll try to speak slower. Yeah, I'll speak slower. So hopefully, more. Um, so, hello. How do you propose we go from here? Is it okay for me to continue or?
Yeah, it's still okay. Okay.
It's
Now it's
okay. Yeah.
Yeah.
Okay, let me try to speak a bit louder and let me try to speak slower as well. This slide here is on the methods that we use a lot in identifying structures. So basically HPLC, NMR, mass spectrometry, X-ray crystallography, and more recently cryo-EM. So Brin in Chile, they actually just recently acquired this cryo-EM machine. So we are fortunate enough to have gone with Brin starting this year, and we are hoping to use IBM a lot more. I know that with Erlangga also just the NMR machine. When I went to Singapore last year, I met the company. Okay, next one. So this is going to be, so that was the introduction. On this side, I'm going to briefly talk about the pharmacology. I'm going to briefly talk about drug targets that we need to cover. So the drug targets that we need to cover are vascular receptors, enzymes, transporters, and nucleic acids. What I want you to be able to get is where in cells are these located. So the enzymes are typically intracellular, they are typically in the cytoplasm. The receptors are typically on the cell membrane. Right? So depending on your view, so if this is a lipid bilayer, your proteins, your receptors, on the GPCRs or channels, they tend to be located just on the outside of these extracellular membranes. Transporters, as it implies, it has to be able to access both extracellular as well as the intracellular sites. And lastly, if you want to target nucleic acids, which is often the case for antiviral compounds, you need to make sure that your compound actually goes to the nucleus. Okay. So for us, especially at the undergraduate level, the location, the structure, and function of your drug target is very important. Right? You need to be able to understand, at least at fundamental level, where your drug is located, what does it look like, if there is any cryo-EM or the X-ray is a structure at the EGD, and lastly, what does it do functionally. Now, from there, typically what we give to our students is do you know the target of your drug? Right? So this is what we want to know. Is the structure of our target known? If you don't, then typically people do what we call a phenotypic-based discovery. So in a phenotypic screen, you don't know the target, but you know what sort of function you wanted from your compounds, right? So for example, in a phenotypic drug discovery, because you know exactly, let's say, I want this compound to be able to inhibit the growth of this virus, right? That is my phenotype that I'm looking for. So then you can develop an assay that looks at exactly that. You then do screening, and then you can identify some hits and leads. And once you get to that stage, then you do some sort of target deconvolution. So either some SPR or some sort of X-ray crystallography, X-ray co-crystallography, or IOBM, so that it allows you to get to the target. Alternatively, on the target phase of discovery, you already know the target, and then from that point onwards, you are developing assay so that you are allowing screening of a high number of molecules to target that initial receptor. So again, drug discoveries in antibacterial and antiviral, they tend to start with phenotypic. For cancer, they typically start from several combination phenotypic phase drug discovery. Finding versus functional assays, this is another key concept as well that needs to occur. So finding, just be aware, it looks as to how tight or how loose the binding strength is between the drug and the receptor, right? Nothing else. So it purely looks at how complementary the functional group interactions are. But with the functional potency, you are looking at the ability of that compound to elicit a function. For example, does it improve calcium signaling? For example, does it actually stimulate cyclic AMP production? So finding not necessarily correlated with functional, but in general, they tend to. Um, Given that my audio is not too good, I think I will skip this activity if that's okay. But if you guys are interested, you can take out your phone and you can scan this QR code. This is basically on an article that um, that is on 5HT to receptor pharmacology. Now, for chemistry understanding at this stage, it is important that when we think about binding and how a binding of a drug goes to the intended targets, there is this process that we call an induced fit. And that induced fit in turn leads to conformational change, and then that leads to the activity that we are looking for. Right? So at the molecular level, right? So if we look at it closely, when you get drug molecule, right? So you take it in a zoom like this. Remember, your drug molecules already have functional groups embedded within it, right? Your carboxylic acids, your amines, your amides, your esters, and so on. Right? So imagine that those functional groups are going to be trying to position themselves so that they are maximizing the intermolecular forces or intermolecular interactions with the intended targets. Right? So as the drug tries to maximize the binding with the macromolecular target, it might not be clear from your from where you are, but this initial shape of the macromolecular target is actually not perfectly rectangular. It's actually more like a trapezium shape. However, once the drug actually binds, and notice that there is a slight conformational change. That very often that slight change in the conformational structure is going to be the difference between a healthy and a diseased patient. Right. Now, when we look closely as to what sort of intermolecular forces that can happen, these are the typical intermolecular forces that I'm sure in high school you would have covered it. So anything from ionic bonds, covalent bonds, ion dipole, ion dipole, hydrogen bonding, as well as. So please make sure that you guys remember these, and we will further apply this when we actually go through the sort of the second half of this lecture. I don't think this audience needs any reminding as to what intermolecular forces are. So just remember, if you have an ion with a dipole, and you have ion dipole effects, hydrogen bonding, like what you see in alcohols and water. Dipole happens with compounds that have dipole moments. Ion dipole is when you have a um, an ion and then some sort of hexane, in this case, double is double, or dispersion, which can sometimes be lumped together with other loss forces. I will not talk too much about this, but hydrogen bonding donor, just be aware the donor must have the OH or the NH. And the target in medicinal chemistry, we normally look at only the nitrogen and the oxygen atoms. Yes, technically fluorine is also a hydrogen bond capable, but we don't tend to put too many. We tend to put fluorines also to cover for metabolic liability. And then from there, on the protein side, so again, you should have done this in your first semester, and that is on protein structure and function. So remember, primary structure is your amino acid sequence. And these days, if you have your amino acid sequence, AlphaFold can easily predict your secondary structure quite readily. So that shows how far relational biology has become. Secondary structure, obviously, again, can be predicted from the amino acids. So this is when you have alpha helical, beta sheets, or even the disordered proteins like in the Parkinson's. Although at this stage, we normally don't ask our undergraduate students to memorize or to remember all the 21 amino acids, generally it's a good point to remember that there are 5 amino acids that are ionizable. So lysine, arginine, and histidine, these are the basic amino acids. Phosphate and glutamates are the acidic ones. And in the second group, I generally put it as the hydrophobic amino acids, so the non-polar ones, so they tend to fall inside. And lastly, those amino acids that is not quite ionizable, but they can form some sort of hydrogen bonding, as well as double. So typically, the ionizable amino acids is going to be very crucial because it can contribute to much highly energetic ionic bonds. Okay. Oopsie. Do okay. And of course, the tertiary structure. So this is typically what you see when crystal structure is solved. So in this case, this is an example where you can see the transmembrane domains from 1 to 7. You can also see the sulfide bonds that give rise to the structural characteristics. You can also see the tendency of the hydrophobic amino acids to fold amongst themselves and to create these more hydrophobic pockets. So a lot of the drug action to proteins are intended to go towards inside pocket of this protein to further disrupt the binding. And finally, with quaternary structures, I just want to put this for completion. When you have two or more subunits, for example, with ion channels, with nicotinic ion channels, you actually have 5 structural subunits that they can actually form. They can open and they can close to allow movement of ions in and out. And also, we also have what we call post-translational modifications. So just be aware that proteins are very dynamic. On the outer side, it's hydrophilic. On the inside, it tends to be hydrophobic. But those outside amino acid residues can then be further prone to further modifications, acetylation, putting on sugars in glycosylation, phosphorylations, carboxylation, etc. And so the process of these PTMs or post-translational modification is obviously very crucial, not only for metabolism, but also for duplicate of those compounds. Now, unlike the first one, I would like the students, if that's okay, can I please get you to take a snapshot of this and open it in your tablets or open it in your phones? I think this is a good practice. So I'll give you a couple of minutes. Please take out your phone. You should be able to scan this, and it will bring you to a PDB code of 63. Okay. And in the meantime, we'll call this Uni. Is my voice okay or?
Okay, Dr. Hendra.
Okay, thank you for
Yeah.
this. I do apologize for these technical difficulties. Can everyone see the PDB screen? Yeah. Okay.
Yeah.
Now, if you go down here, you can use this on your phone as well. Please look at this one. So explore in 3D, and you can click structure. Right. So from the top there, when you see this cartoon of the protein, click on the structure. It might be a bit slow, and depending on which one or which tab that you use. But this is a really nice view. So on the top there, this shows you the amino acid sequence. And when you scroll down, so if you use your tablet or your phone, you can actually use your index finger, and you can actually do this um, with using your touch. So what this does is typically it allows you to see that drawn in the alpha helices, right? So these alpha helices, these data sheets, as well as these intrinsic plates of the proteins, those terms, those twists, that allows you to see in 3D what that protein actually looks like. And then hopefully when you rotate enough at the right angle, you will be able to see a small compound, a small molecule in this case, which is a parent, and they are actually bound to your PAP1 protein in this case. And if you click on that once on the computer, then it will automatically zoom in and it will tell you what sort of electromagnetic forces you are actually looking at. Right? So, for example, on this ring, you are seeing a aromatic stacking with this tyrosine structure here, and then you can do a rotation. So here, that is the again stacking between this part of the molecule and again this tyrosine. If you scroll here, that's a nitrogen that is going to be charged. So that's going to be interacting with this carboxylic acid down here, as well as doing some hydrogen bonding. Right? So these are the kind of information that is useful for us when we are designing drugs or when we want to further optimize the physiochemical properties of this molecule. Right? So if we know that this compound binds, this compound achieves the function that we wanted, but for example, let's say it suffers from being too lipophilic or being too lipophobic, then we can do changes. But we're not doing those changes blindly. We are doing the changes to certain areas that we know is not going to disrupt the important bindings that we see. Right? So, for example, here, that nitrogen here is obviously very crucial for the ionic bonding. So we can perhaps make a bit more extension. Let's say, sorry, and let's say my light had turning off automatically. So and likewise, if you look at this position here on the transactional indole, that is actually further space that you can exploit, right? Again, for example, there is this histidine that you could potentially use to connect to your small molecule. So and obviously, in the PDB bank, there is a lot of this structure that has been published. So when your lecturers is going to ask you how you understand your drug and protein interactions, it is not unreasonable that they will show you a diagram and they will ask you what sort of modifications are they going to propose to make this compound, I don't know, more water soluble or less lipophilic, or less metabolically liable. Okay. So please play around with that. I think that's the one thing that we are telling our current students here. This is typically at second year level. We tell them, look, let's look at this. We don't need you to memorize stuff. It is more about applying what you've learned so far and identifying in 3D what could be changed and what should not be changed. Right. So now I'll go back to my screen. Screen two. Can everyone see slide 22 now? If I can get a nod.
Yeah, we can see,
Yeah.
Dr.
Thank you. Thank you. So that was a bit of a discussion on the receptors. Again, how am I going with time? I'm almost there. So look, I will skip this. There is this discussion on slide 23 onwards. This is more about when we discuss neurochemistry, when we're looking at neurotransmitter signaling. So I'll go through this very, very briefly. But on the nerve signaling, your presynaptic receptor sends in either your dopamine or your serotonin, right? That's some sort of messenger. That messenger is then going to engage with the receptor that is in your post synaptic cells. And then what happens to that is basically, again, that binding of that messenger to that receptor, that induced fit is going to happen. There is going to be a slight conformational change. And that slight conformational change is what's going to be responsible for the downstream signaling that we are seeing, for example, when people take other antidepressants or anti-manic, anti-schizophrenia, and so on. Right? Mental health is very important, and I sincerely hope that everyone doing this class is also taking a good care of their well-being. Right? Don't worry about this slide. Again, the intention here is to let the students know that the chemical messengers, they tend to be short-lived. So once they are in the synapse, they do the job, they bind to the receptor, they activate the downstream signaling. Typically, then the messenger has finished its job, and it goes back, right, and gets recycled for the next step. But that induced fit, right, as we talk about, although seemingly only minute in terms of appearance, but this nice diagram shows us that before the binding of the drug, these two positive and negative charges were a bit too far from each other, right? But after the binding of the messenger, then you actually bring those two much closer, right? So you are further improving these intermolecular forces, and this is what gives rise to the slightly altered shape of the protein. Ion channels are similar. As I said, when you have more than 2 or 3 subunits, then you can form some sort of tunnel. And depending on the messenger's ability to activate or deactivate, then you can sort of open or lock these ion channels. Right? So these ion channels are very prevalent when we talk about GABA receptors, when we talk about GABA 8 modulators, positive allosteric and negative allosteric modulators. Don't worry about the nicotine. GPCRs, again, this is just meant to be very briefly. All we wanted to tell the medicinal chemistry student about the GPCR is that there's a transmembrane. They are coupled to this G protein structure in the intracellular site, and GPCRs represent a massive target for pharmaceutical industries, right? Opioid receptors, glutamate receptors, GABA receptors, these guys, they're all GPCRs. Right? And so on. Look, I'm not going to talk so much about this. I think this is too pharmacology heavy, but I think it shows you that there is a lot more pharmacological context that needs to be understood when we are looking at and designing the functional assays for GPCR modulators. Right? I wouldn't worry too much about this. Typically, what I tend to tell my chemistry students is that you have different G proteins. Those G proteins have very different functional activities. So it depends on what you're looking at. So if you're looking at FAP-HT2 receptors, for example, you're typically looking at the calcium signaling. Or if you're looking at others, then you can look at cyclic AMP production, like in cannabinoids, and so on. So that's on the receptor side. On the enzyme side, I think we're a bit more familiar. So when we try to design enzyme inhibitors, typically we talk about how we can mimic the transition state of the substrate. There is also a difference between orthostatic and allosteric inhibitors, which hopefully has been covered previously. So just to recap, review very briefly, orthostatic means that your inhibitors actually binds to the same site as your substrate, whereas allosteric actually binds to a different site compared to your substrates. Right? So in the antiviral drug discovery, in the antibacterial drug discovery, a lot of the antimetabolites are actually used to disrupt functioning of certain enzymes. So again, this is what it looks like. So when an enzyme processes the substrate, again, there is that induced feed process to speed up product. And obviously, if you want to design an enzyme inhibitor, you want to mimic what the substrate looks like, but effectively you are blocking this process. So whereby your inhibitor will simply occupy the binding site of your enzyme and hence blocking the access of your substrate. So the product is never produced. This is an example, again, step by step, more on the biological chemistry mechanism. So in this stage, you are doing a hydrolysis of a peptide. So in this case, you have a triad of serine, histidine, and aspartic acid that they work in conjunction. And so as you can see, the serine initially do an nucleophilic addition to form these tetrahedral intermediates. The histidine then takes up one of the proton to make it protonated, and then from that point it does a nice ionic bond, so that's very much stabilized. And then at that point, water molecule comes in, and it does the hydrolysis. So the your protein with the other side of the protein completely. On these acid and active transporters, look, I'm not going to talk too much about it, but for example, in diabetes, we have sodium glucose cotransporter inhibitors. So in that case, you are basically looking at in certain diseases, there is often a overactivity of certain transporter, and our job is to make sure that that transport process is being modified. We normally tell students, and there is a difference between passive and active transport. So the passive transport is literally just passive diffusion. So it just goes following the normal concentration gradient. So if you, for example, administer a drug, you're initially having high concentration on the extracellular side, but as time goes by, just by diffusion, your drug is going to be able to go towards the inner side. The active transport, on the other hand, requires energy, and this may or may not follow the concentration gradient. So, for example, in Mycobacterium tuberculosis, there is this ATP synthase enzyme that we can actually target in order to come up with a newer NDTB drugs. And so finally, with the drug-like properties, so it is our intention that students at this level can start seeing the connection between the structural properties, right? So for example, your molecular weight, your hydrogen bonds, your lipophilicity, your polarity, your pKa, the shape of the molecule, as well as the reactivity. Right? So this is your fundamental organic chemistry that you should have kept by now. And being able to slowly build up sort of these physiochemical properties on the solubility, permeability, chemical stability, as well as more on the protein or the biochemistry side, the metabolism, the binding, and the affinity. So a combination of these physiochemical properties as well as these biochemical properties will ultimately influence how good you are when you are given as a drug in a living system, right? For example, how stable are you? How bioavailable are you? Is it quickly cleared? Is it too toxic? And so on. Yeah. So about this, this is more about just for us to see it in a bigger picture. As I said, when we do drug development, we typically work in a big team, and chemists tends to perform these three, right? These three sections, and or actually more like the middle two, more like the middle two. So just be aware that it is not a linear process. Medicinal chemists will make, I don't know, 20 compounds in 1 month. We give it to the biologist. The biologist do the screening for us. They then come back saying compound 1, 2, and 3 is good, but 4, 5, and 6 is not so good. So we then take those feedback and we try to explain why the result is the case, and we are going to further improve our compounds. So yeah, that's on the acne talks. So I think that's probably enough on the initial sort of overview of the physiochemical barriers, the kind of drug targets that we need to see. The last sort of half an hour or so, I'm going to talk about the chemistry aspect of it. Right? So we're on slide 41. Look, I do have about 80 slides, but I don't mind if I have to finish a bit earlier, or I can go through this slightly faster. Right? Again, the intention is for me, not for the students to try to memorize or what did Hendra talk about. It is more about, if I give you a compound, right? If I give you 2 or 3 structures, I want you to be able to see what changes did the chemist do. Why did the chemist actually do those changes, right? So that basic understanding is actually more important at this stage. Okay? So just to recap on what the chemist does. Once the target disease is identified, we know what the drug target looks like. Assay has been developed, right? And slowly we're finding hit compounds. Then, as a chemist, we are tasked to improve not only the activity, but also the physiochemical properties of those compounds. And so this is the heart of that process. At the end, once you do enough structure activity relationship, the idea is to hopefully identify a pharmacophore. And I will go through what pharmacophore is, and then further lip optimization before going to more advanced or even ready technologies like the toxicology, safety test, genotoxicity, and scale up, patenting, IP, and so on. Right. So what do we want to do? Well, what we want to do at this stage as a chemist is we want to know which functional groups are actually important for the binding and or the functional activity. Right? So in terms of the workflow, we will look at one functional group at a time. We will try to change it to something else. We might try to remove it. We might try to mask it, right? Whatever you do. But make sure you do one change at a time and you are seeing what is the feedback. Does that change of that one functional group actually bring better, no change, or worse activity? Right? So this is what you wanted to see. So after you do the testing, you then do some sort of conclusion, depending on the method of testing, whether or not that functional group change that you did was actually beneficial or no good. Right? Depending on what assays is available at that moment. So typically in vitro is easier to set up, right? So as I said, if you do not know what the target is, typically endpoint measurements, for example, does it kill does it kill the virus or does it kill the microbe? Right? That could be your endpoint measurements. But if you have a binding assay already set up, typically involving radio labeling, and you can see how much of the radio label substrate actually gets composition. So at the end, it is these 2 points at the bottom that is important, right? If the in vitro affinity drops because you change that functional group, that means that functional group is important for binding, right? However, if the activity is actually not affected, that implies that you can further do modifications. Right? So seemingly quite easy to understand. What sort of modifications can you do? Well, in chemistry, in organic chemistry, you cannot run away from changing the statics or the electronic factors. So statics is more about the size, how big, how small, and electronics about electron donating and the electron withdrawing capabilities. So the easiest, for example, will be to do what we call a me-too analog. So you can change a naphthyl to ethyl to propyl and so on. Some of these analogs, when we decide to do what changes, we need to also be cognizant about how difficult the chemistry is going to be, right? So, for example, there is no need for us to do a much more difficult, technically, the chemistry modifications if all we want to do is to simply probe how big the hydrophobic pocket is, right? It can be challenging to actually, for example, replace an aromatic ring with a cyclohexane ring. So you do want to be strategic with how much resources you want to spend to get the same information that you want. And once you go through all this process and through process of trial and error, you will end up with some sort of understanding of your pharmacophore of your target. And so a pharmacophore definition is given to you here. So it is a collection or an ensemble of steric and electronic effects that is needed for the ligands or your drug molecules to bind effectively to the intended target and elicits the wanted responses. So for example, let me go through this. So can I please get some response from the chat box, if that's okay, right? So if we look at this morphine on the top, morphine is obviously highly potent painkiller. If I replace morphine with codeine, right, there is this methylation of this OH group on the top. Activity drops quite significantly, as we know, right? So codeine is only about 20% activity compared to morphine. And obviously the codeine is meant to reduce the morphine dependency. So in this case, can I ask the audience, so is this OH group good or bad for the painkiller activity? Good, very good, right? Thank you, Natalie. I hope everyone else is also the same. So as you can see, right, so one change at a time, and you're seeing a drop. So that means the morphine, without all the alkylation there, that's the most potent, right? You might be wondering, but if that's the case, why do we do codeine? Well, remember, we are doing codeine because we know that the morphine is too addictive. And that is why, in this case, we are deliberately toning down the activity. Right? So remember, everything has a purpose. So you are trying to work out exactly which part of molecules you're changing. Second question, please. Now, if you look at the bottom OH group, right, this allylic alcohol, I then choose to oxidize it. Right? So I can choose to oxidize this because it is an allylic alcohol, and I can do a chemoselective transformation so that I don't touch that phenolic alcohol and I'm only oxidizing. Now, when I do this, the activity is actually unaffected. So what does this mean to you? Is this OH group important or not really? Not important, right? Or at least not important enough, because when we change it, right? When you change it, that OH group, you might be telling yourself, okay, what sort of intermolecular forces can you do with this? Well, Andra, obviously it can do hydrogen bonding, right? Obviously it can be further alkylated, but it seems like when you oxidize it, you actually don't need, you actually don't get anything. So depending on what the need is, you can actually choose to perhaps eliminate all these OH group completely, or you can think of it as a potential site that if you want to further extend your drug molecule, then this is a good spot for you actually to start, right? It is a bad place to start extension on this OH group because, as you can see, the codeine activity actually drops quite significantly, right? So hopefully you get the gist, right? So change one thing at a time, what the activity looks like, change one thing at a time, do what the activity looks like, and keep repeating the process with each of the different processes. More example. So this time, it is about making the compound going smaller, right? So look at the morphine. Yeah, this is the same example. Now, pay attention to the ones that we label in red and what we label in yellow. Right? So notice, if you compare morphine and metazazine, you'll notice that this ring is gone. Right? This ring is gone. So effectively, you are deleting all this stuff. Now, what if I tell you that metazazine is actually about 2 to 5 times more potent than morphine? Right? So in this case, structural simplification not only makes the compound similar, right? The compound still maintains the pharmacological activity that we want, but we're making this compound more atom economical. Right? We're making this compound already achieve what it's intended to do without, using too much unnecessary atoms. Likewise, if you then compare metazazine and orthogonal, you sort of recover that ring at the bottom, right? And I can also tell you that metazazine and orthogonal again have fairly similar activities. Right? So from these metazazine and orthogonal studies, we can actually conclude that it seems like as long as you have a phenolic ring, right, in red, and some sort of 1, 2, 3, 3 carbon-carbon bonds length away, and then you got some sort of ionizable nitrogen, it seems to be the basic structural characteristic for you to actually achieve anesthesia or painkilling. Right? So that's how, through a concept of morphine, we can actually sort of shrink down what is the minimum skeleton that is required for a certain activity, which is painkilling in this stage. Right? So once we have obviously a lot of analogs being produced and we are seeing what the profiles are, then we start to see some sort of summary that looks like this. Right? The phenolic group is important groups. That nitrogen is absolutely crucial. Right? So that is my basic skeleton. It defines the minimum skeleton. And so by implication, this oxygen ring is actually not crucial. Right? So is this OH group, and so is this double bond. Right? So if depending on the availability of your starting material, and depending on what sort of pharmacological profile you want, you can actually start thinking about new opioids using these fundamental characteristics. Right? So on 3D, this is what the T-shape of morphine typically look like. And again, this is just to show you that if you look at the basic pharmacophore, as long as you have aromatic ring, the phenol, and certain distance, right, to a nitrogen, then you can come up with this sort of 3D representation of the pharmacophore. And you can do, when you do modeling, when you do computational studies, you can actually design molecules in silico, and you can see whether or not your proposed compounds actually fit the description or not. There is no point to make the compounds that doesn't fit the description. So what I described for morphine just now, you can do the same pretty much with other CNS or brain neurotransmitter. So again, for example, in here is the dopamine. So this is the basic structure of the chemical neurotransmitter. And so again, if you think about dopamine, you think about this aromatic ring. You do have 2 OH groups, and again, 2 carbon-carbon bonds away, you have your basic ionizable nitrogen. Right? Now, if you want to think about dopamine analogs or dopamine inhibitors, dopamine antagonists, dopamine superagonists, depending on what CNS indication you're trying to cure, you can think about how flexible your dopamines are. Right? So if you think about this carbon-carbon bond and this carbon-carbon bond, they're actually quite flexible. So they actually can rotate in 3D. So once we know what sort of conformation we want, so for example, in structure 1, we are drawing a second cyclohexane ring, connecting it to this carbon, or possibly conformation 2, whereby you are locking the second ring relative to this OH here, then you can actually test analog 1 and analog 2 to see whether or not they're actually more or less potent at which dopamine subreceptors you are trying to examine. Right. So, how am I going with time? 11:20. In my last 10 minutes, or maybe in my last 10, 15 minutes, let me go through the lead optimization strategies. Right? So this is often the bread and butter that we are trying to give our second year chemistry students. Right? So there is 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 strategies are what's commonly shown in publications. When you look up Journal of Medicinal Chemistry and other related journals, you normally don't run far away from these 9 typical variations. Right? So in my last 20 minutes, 15 minutes or so, I'm going to show you examples. Right? I want you to look at the structures from the beginning and what they propose, and hopefully you can see what they're actually trying to do. Right? So first one, probably one of the easiest to understand is to vary the alkyl substituents. Right? So in this example, what do you see? You see a methyl group, and you are trying to see the much bigger, more lipophilic third butyl group. Right? So on the left-hand side, let's say your lead compound has some initial activity, right? But let's say when you change the methyl to third butyl, in this case, right? Let me ask this audience: do you expect the compound on the right, this compound here, do you think this will be more active or less active compared to this one? More active. Well, Natalie, you're obviously have been listening. So obviously in this case, again, Natalie, you're right. Since it actually, as you said, it does more van der Waals interactions with this hydrophobic pocket that we're trying to locate. So in this case, you strike lucky, so you make them bigger, and the bigger the pocket at that stage, actually the more van der Waals that you can actually get out of it. So in this case, yes, compound on the right is going to be more. Now, let's look at distance effects. Right? Now, receptor 1 and receptor 2, consider this as 2 structurally related proteins. So, for example, dopamine type 1 and dopamine type 2, or serotonin type 2A versus 2B, right? So, 2 receptors fairly similar, but one tends to be bigger than the other. Right? So, for example, here, right, if you have 2 compounds, one with the N propyl group and one with the N methyl group. As you can see, because receptor 1 has more space on this top right-hand side corner, it doesn't matter whether you put N propyl or N methyl, you will see some sort of activity. Right? So in other words, N methyl or N propyl does not actually have a selective activity for the receptor. However, on the receptor 2, right, when you have a slightly smaller on this side, notice what happens at receptor 2, the smaller N methyl group will still bind nicely, right, because methyl is not capable of causing some steric block. But this compound, the N propyl, is going to have a hard time receptor 2 because there is some steric blockage here. Right? So for this example, when you do selectivity studies for N-methyl compounds, you're not going to be able to see any selectivity because they're basically defined in a very similar manner. But if you find the N-propyl will bind better to receptor 1 and bind less to the receptor 2. Right? So in other words, if you have an N-propyl analog of this, you start introducing some elements of selectivity for receptor 1. Right? Now, why is selectivity important? Well, obviously, because often diseases and often certain abnormalities is only caused by just one subtype receptor problem rather than the overall things. Right? So in this case, the beta receptors versus alpha receptors, you can actually take advantage of that difference. Now, obviously, in terms of the synthetic feasibility, this is when your organic chemistry knowledge comes in handy. So, like what Uni initially said earlier during the introduction, combining your synthetic chemistry knowledge as well as your medicinal chemistry, it really goes hand in hand, right? So, from MedChem studies, you know exactly what you want to achieve, and you are using your synthetic chemistry, your knowledge about the activity of those functional groups, and change it to the desired targets. So, for example, with the ether, you can do hydrogen bromide treatment to do a O demethylation or dealkylation, and then followed by strong base, sodium hydride that has the pKa to deprotonate that hydrogen of the primary alcohol, and then you can do subsequent alkylation chemistry with iodides. Again, alternatively, with an ester structure, you can do a basic hydrolysis. Right? So depending on whether or not you have the alcohol side, the carboxylic acid side, and again, you can further form different esters by treatment with acetyl chlorides, or you can do further fixture aggregation with different alcohols. So that's the split outline, right? Variation of the alcohol, move on further. Variation of the alcohol substitutions. Now, this is what we call the aromatic wall, right? So you go from position 2 to 3 to 4, right? You go from alcohol to methyl. So obviously, as you can see here, we have a para substitution, and that's always true. The phenol is having not so well. It's not so well aligned with the hydrogen bond receptor on the skin. So, right? But when you put it at the meta position, then that OH group is positioned correctly. And so, in this case, obviously, the metal substitution again has a closer exposure to the intended side. And so, obviously, metal here is going to be more. And as well as this audience, we would like to ask what happens if I move it to the ortho. So do you think this will be less or more active? Hopefully it will
I'm
be
sorry,
less, right?
Dr. Hendra, we cannot hear your voice.
Oh, okay.
Yeah.
It's okay. So if after that it is always group is
okay.
placed on the ortho side, then that would be even less active. Right? And on this one is the electron donating versus withdrawing and resonance effects. So again, this is influencing what sort of functional groups that you want to put in. Right? So, for example, the electron withdrawing group, such as a nitro group, is going to be stronger when it's actually at the para position. Right? Because at the para position, not only inductively you are electron withdrawing, but also by resonance, you are getting more structures. So if you actually want your aromatic ring to be very electron efficient, and putting a natural group at the bottom position will lead to extension is probably something that is reasonably easy to understand. So again, conceptually, right? Look at this. The drug, let's say, has initially three functional groups, and then there is the last one. Right? There is this last one. You saw there is an unused amino acid residues, and so you can foresee that if you put an extra functional group that can capture that extra interactions, then you should be able to improve your efficiency. So that's exactly what happened here. So on slide 59, again, this is the pocket, and you can see on the left-hand corner here, there is a hydrogen pocket. So by putting the CH2 final group, you are able to fill binding pocket. And so this compound here is going to be more active at inhibiting ACE and so on. An example of extensions is more dramatic. So in this case, you actually have a nut gas in this organophosphates in tyrant. And if you do some sort of extension and putting in some endogenous acetylcholine-like molecule, you come up with a medicine. Right? So if you look at the extension process, what you are adding is these two extra carbon. You are adding this ionic bond, so you are mimicking the acetylcholine, right? And so what happens is when people are having sudden exposure, their acetylcholine and cholinergic system is going to be affected, and so they need to be treated with the chemotherapy. More example on the extension, again, you can see very different profiles. So, for example, you can change adrenaline, which is an agonist, to an antagonist. Right? So you start from something that activates your receptor into something that blocks your receptor. Likewise, with histamine and the antihistamines. So the histamine is going to be what's causing the itchiness, right, in your skin. And some other thing, obviously, is anti-ulcer, and that's what you actually reducing the histologic transmission. Right? Extension can also come in flavor of chain extension or chain construction. So in this respect, you're kind of seeing the compound in two blocks, if you like, A and B. And what you're doing here with the chain extension is to simply either you lengthen the distance between A and B, or you shorten the distance from A and B. Right. So an example here is again on the expansion. So for example, on the left here, you got a tricyclic 6 system, and when you expand to 676 system, you'll notice that this second 600 ring is now having a better overlap with the hydrophobic interactions. So in this case, the ring expansion of 676 actually works better. More example of the big expansion. So again, on the left, you get a 6-6 system. On the right, you get a 7-6 system. And as you can see here, by putting the 7-6 changes, you are forcing this carboxylic acid, right, or carboxylate that is initially on this side to be having a better overlap with the ionic bonding regions. Right? So these are the kind of changes that we can do to further improve our ring variations are more typical when you are looking at patents and bioisospheres and isospheres. So as you can see, so this is very popular in the 50s, 60s with the NSAIDs. At the time, we know that you only need a heterocyclic or scaffold, and as long as you have two aromatic rings, then typically it has some sort of NSAID activity. So you can imagine you can start with alphines, you can start with pyrazoles, benzene, and cyclopentene, and so on. Right? Other example of ring variations, phenyltropidine, again, quite common. And then in this case, you're taking out the acetyl group and putting in a cyclopropane. Another example would be if you look at methylene or noradrenaline, and then moving to alcohol. Right? So what you are seeing here is the ring variation. So you start with a mono a catechol system with just one benzene ring, and you go to this sort of naphthalene or a bisulfate structure. After this and process this again, I'll touch this very briefly. After stage by definition is a functional group with that has the same valency. Right? So, for example, hydrogen and fluorine, they both can be considered associates, OH and NH, carbon and silicon, nitrogen and phosphorus, right? Oxygen and sulfur, and so on. Whereas bioisosteres, they tend to be not exactly looking very similar, but the overall activity is actually maintained. So it is much harder to actually spot isosteres compared to just isosteres. So there's a lot to cover here, but hydrogen and fluorine, hydrogen and deuterium, NH&OH, ONS, chlorine, bromine, iodine, carbon, silicon, carbon double bond, carbon hydrogen double bond, carbon oxygen double bond. So and an ester going to ketone, amide, bioamides. So these are your typical bioacids. Right? So for example, here in this example, slide 70, if you have OCH2 group, right? If you want to replace the oxygen with an alkene, right? Or you bring oxygen with sulfur, and you put the CH2CH2, it eliminates all the activity. So in this case, that simply means that the oxygen is actually important for some sort of oxygen bonding receptor. And if you're trying to eliminate that, then you disrupt activity. And again, if you replace O with NH, that retains some sort of activity. So again, that supports your conclusion that there is some sort of hydrogen bonding interactions happening at that region. More example here. So this one is the bioisosphere. So if you look at the amide structure here, and as you can see, we now incorporate another ring. So it's pyrrole ring. So this pyrrole ring is considered a bioisosphere for an amide group. So if you look at the rest of the molecule, they're actually still the same. So they're maintaining everything else. They just do what we call a scaffold hopping. Right? So scaffold hopping simply means you keep the other two things or the most things the same, and you just change one sort of scaffold at a time. So in this case, it's an amide to a pyrrole, and in this case, you actually get some selectivity improvement. Likewise, I don't have time to go through this, but if you have a carboxylic acid groups, then you can also look up tetrazoles as your bioisosteres. We're almost there. So simplification, I will not talk too much about this. You've seen the examples before in morphine. The idea is that we want to be atom economical. If we can achieve the same activity with less atoms, then that's good, right? So this is the cartoon representation. So for example, if the initial drug has the 6 functional groups, we can actually chop down, for example, the carboxylic acid, the chloride, and the toxic group. And we're down to these 3, but they actually are still maintaining activities. Then obviously we have a much more atom economical drug structure, right? So there is a lot of benefits from having a simplified structure. So you are more likely to satisfy your Lipinski's rule of 5, and there is more chance for you to control your metabolic liabilities. Right? We've seen this example before, right? So morphine going to laforphenol, going to metazazine. So if you like, we are chopping off this ring in middle to get the levorphenol, and then again from levorphenol you chop off that ring to get metazazine, and as you go from morphine to levorphenol to metazazine, you actually get a slightly improved activity enhancement. And word of caution, so it doesn't mean that you should prune or you should just cut everything straight away. You need to do the cutting in stages to avoid the oversimplification. Right? So for example, at some point, if you decide, oh, let's cut this nitrogen group off, then you will see basically a compound that is non-active. So you want to do the simplification at stages. It is obviously important. It can be time and resource saving exercise, especially when you have a functional group that is asymmetric. So it can have R of S stereochemistry. But just be careful. Although it is tempting to cut all those asymmetric centers. Very often, the challenge would then be that your compound is going to be too flat and you don't have that 3D sort of shape recognition anymore. So just be aware, you can save money, but you can have very limited chemical space. And lastly, rigidification. So this concept, again, I've kind of touched upon this when I talk about the dopamine. So the concept is to look at if you have the flexible part of your molecule, can you actually lock it in to a certain conformation? Right? So, for example, in this example on the left, upon single bond rotation, you can have sort of this cis against cis, or more like a trans, or more extended conformation, right? So out of these three conformations, which one is actually the relevant one that you actually wanted? So again, in this example, you can do rigidifications on slide 77. So in this sample with the compound 1 going to compound 2, rotation wise, you notice that in the conformation 1 that you have a much better ionic bonding here, but with the receptor 2, in which you have a slightly different positioning, this conformation 1 is not going to work so well when you want it to bind the receptor 2, right? And so depending on which receptor you want your compound to be more active in, so for example, if you want it to be in receptor 2, then you can start to do some rigidifications, typically by introducing a ring structure, right? So again, you can introduce a ring structure between this nitrogen and this carbon here to make it more rigid, or likewise, as you said before, you can take it from this OH group to this carbon position. Right. Again, more example, metastatin. You can start with the sortable bonds, and you can do a dehydration chemistry. So you can change it to a Z isomer, which is more cis, and the E isomer or trans, which is more extended. Right? So in this case, it turns out that the cis isomer is actually more active. So that means this conformation is what's more beneficial. Right? So there is no point to actually do the E isomer. Let's change it to the Z. Not only you remove this metabolic liability, but also you are getting extra activity. Right. So more example here that I'm not going to talk too much. But again, the idea is the same. If you have a flexible side chain, you can introduce an ortho methyl group. So you're deliberately introducing some steric clash. And this strategy is often seen when you are looking at kinase inhibitors. So for example, on this one here, without the methyl group, these two panel rings are pretty much coplanar. But once you install the orthomethyl group, then you start seeing this more orthogonal formation. And that is pretty much my last slide, everyone. So typically, I give this lecture for the first 2 weeks of our medicinal natural products chemistry unit. And then from there, just to let everyone know, typically from there we then go to specific drug groups. So the next one that normally I cover is the antibacterial drugs, and then I normally went to antiviral drugs, and then I go to inflammation, and then go to adrenergics, CNS.
Sorry. Yeah.
Yeah. Okay. So look, Bu Yuni, I'm Putri, I'm Pukolis. I'm happy to stop here. If there is any questions, or if there is anything that is not clear, please let me know. Thank you again for the invitation.
Yeah. Yeah, thank you Dr. Hendra for very untuk presentasi yang sangat membuka wawasan gitu ya. Sebagai informasi ini anak-anak kami ini baru saja menyelesaikan kuliah topik organohalida. So I hope that now you know why you learn stereochemistry ya. Kenapa stereokimia itu menjadi sangat penting. Jadi baru saja ya minggu lalu hari Jumat lalu terakhir sampai di organohalida untuk organic chemistry-nya. Jadi kuliah hari ini benar-benar memberikan insight pada anak-anak bahwa mereka memang harus bersemangat gitu ya to develop the drug. Okay, I see some question. A student has already dropped their question on the chat box. Yeah, for students, you can ask in bahasa because I think Dr. Hendra also understand in bahasa Indonesia ya dok ya. Okay, or in English as well. Okay, I will read the first question from Bijita. Yeah. Okay. Can I ask question? So, or you can directly.
I think I can directly read it, Ibu, if you don't.
Okay.
I think there is another one from Shakila Nova ya. Shakila Nova first. Just above Brigida. Gimana caranya mengubah senyawa obat yang harul menjadi acarul? Jika diubah menjadi acarul, apakah masih akan berfungsi? Thank you for the question, Shakila. So, what I meant with this, so on slide 75, right? So, this is the carol drug, and you see that there is this asymmetric center that can have R or S. So, when we say we change carol to acarul, we simply change one of these X, right, or deleting one of these H, so that this carbon is no longer possible to form R or S. Right? So, for example, if X is oxygen, right, and then H is hydrogen, Y is nitrogen, I can change X, for example, to another hydrogen. So I can have CH2 groups, so that carbon is no longer going to be carbon, right? Obviously, if you do that, then you are no longer having that chirality. And obviously, chirality is important because depending on what X is, that can go to in three-dimensional way, that can go and access the actual amino acid residues that you want. And sometimes if you delete that, then you're no longer able to get that interactions. So with your questions, if it's acarol, apakah masih akan berfungsi? It depends on the change that you did. If the change that you are doing is not important for the binding, you'll be fine. But if the change is crucial, then you will see a dramatic drop in the activity. Okay, so I hope that helps. Untuk next one, Miss Brigita, drug lab, I want to know how do researchers actually decide when a compound is good enough, right? So good enough / not good enough. Lipinski's rule, Pieper's rules, all there is this like specific rules that we try to teach our students at the beginning. But generally, Brigitte, you want it to be reasonably small. When I say small, is less than 1,000 Dalton. So that's a good starting point. You want it that it doesn't have too many hydrogen bonds, donors. So if you have a lot of NH and OHs, that's normally a bad story. You don't want it to be too ionic as well. So if you have, for example, carboxylic acid and amino group at the same time, then chances are you're going to be a sweet RNA compound and you're going to have a hard time with absorption. So it is predictable. So if the question is it predictable or unpredictable, it is predictable. Normally what we do is I have software that I normally use. You basically put in the structure in there and it will tell you. Very likely this compound is going to be bioavailable or not really. So no, very early on in the lead optimization, we can combine the SAR and the SPR to come up with the best compromise between the two, if you like. Okay, because things that are normally good for the activity, but let's face it, are most of the time not the best for the property effects. So you do need to have a balance between the two. Okay, so I hope that question is answered. Next one, oh, we have quite a few. 15 new messages. Okay, let me try to go a bit faster. Next one, Sophia Vardani. Receptor are classified as a mechanism. How do we determine whether it is suitable? This will we rely on the biologist, Miss Sophia. Okay, the biologist is going to be the one telling us. We suspect that this disease happens because of the abnormalities of receptor X subtype Y, right? And how? If the question is, how do they know that there is this kind of receptor? Omics. So either the genomics, transcriptomics, proteomic studies, and metabolomics. So typically, we compare, for example, DNA sample from disease patients versus the healthy patients, and we're trying to see some polymorphisms difference between the two. Okay. No, chemists do not have that knowledge. We rely on the biologists. Next one, Harriet. In the context of drug discovery, how do we decide whether to use phenotypic screen or target-based screen? Quite simple, Harriet. If you know the target, you start with the target-based screen because that saves you a lot of time down the line. You have a better control over what you are trying to make in your library. Phenotypic screen, you tend to go sort of blindly, right? Phenotypic screen is more useful when you don't have good hit compound to begin with. Okay, so I think with GPCRs, for example, if you know exactly what GPCRs you are working on, target base is definitely more advantageous. Okay. Next one, Ms. Dewi Praja. What's the primary challenges, especially with technological advancements? The primary challenge, I would say, is what to make, right? What to make and what to cure, and what hasn't been done, and how can we make quick impact by doing things, by identifying those things that haven't been done. So in the past sort of 20 years, people have been looking at repurposing, right? You see it during COVID, people try to use that. It seems like the concept of repurposing, although it's good, but it actually hasn't been very fruitful. And then AlphaFold actually came into mind. And then, as you guys may be aware as well, there is more and more AI software that can propose to us, hey, this compound could be useful initially for antibacterial, but could be useful, I don't know, for cancer. Right? So there is more and more of this AI, machine learning technologies. I think the challenge would be, well, which one do you want to cure first, and how quickly or how adept are you at picking up those connections that people haven't seen before. Okay. Next one, Miss Elsa. Miss Elsa, could rigid molecules increase the risk of immunogenicity due to their unnatural, non-flexible structures? Immunogenicity, probably not. A toxicity, perhaps yes. It could be more toxic because if it's in the natural form, you are seeing that flexibility. So if you like, you don't get as potent of an activity. But when you have rigid molecules, because you improve that potency, perhaps you get hit with some sort of side effects. That is possible. But again, Elsa, the rigid molecules, typically we introduce it to introduce some selectivity. So it will be counterintuitive if we introduce selectivity, but we still get the toxicity. Okay. Next one, Delshad Muhammad. Saya pernah baca sintesis nanopartikel emas. Apakah benar punya gugus farmakologis untuk pembentuk nanopartikel? I need to see exactly what article you read, Pak Delshad. Yeah. But in general, gold nanoparticles, all these, you need to be very careful with which journals you're reading. Okay. I would generally recommend my students try to read the Q1 and the Q2 journals, and look at the previous publications from that group. Yeah, and sorry, this question is a bit too broad. Without looking at the actual article, I'm afraid I cannot comment. Next one, Miss Sifa. Drug target interaction at molecular level. Is it possible for a drug to bind strongly but will still be therapeutically ineffective? It's possible. That is simply meaning that the target that you are trying to optimize the binding for is not the right target for the disease that you are trying to cure. Yeah. And next one, Imamul Aziz. How can small errors in determining the structure lead to failure of a drug candidate synthesis? The small error in this case tends to be on the physiochemical property side. So, for example, is the compound too lipophilic? Is the compound not water soluble enough? Is the compound having bad pharmacokinetic properties? If the compound is not as metabolically stable? So this tends to be that small error that lead to the failure of the drug candidate synthesis. So that's why these days, not only SAR, but also SPR, the property aspect, needs to be considered early on before we actually invest the money in doing the scale up and doing the clinical trials. Ms. Livia, is it better on new molecules to achieve more beneficial clinical outcomes? I think it depends on what the focus of the company or the academic group is. If the focus is on like fundamental new molecules, then obviously it is great. For example, astaxanthin, right, have very powerful antioxidant effects. Obviously, that could be useful for a lot of things. For example, for diabetes. But other new completely molecules, perhaps you're talking about from marine sponges or very unique bacteria, right, in the volcano, that sort of compounds. That's also fundamentally very interesting. So it depends on how much resources you have, is probably the answer I'll give. Fiorella, if a compound violates Lipinski's rule of five, they're not necessarily bad. Okay, that's why I'm saying, although it's a rule, Fiorella, treat it as a guidelines. Okay, you can still save that compound by doing systematic changes, as we just discussed. Kevin, advancement in purification. HPLC now has UHPLC. Spectroscopy technique NMR has like 900 megahertz NMR. So it's quicker, faster, more expensive as well, obviously. X-ray now has cryo-EM. So yeah, look, technology will keep up. Question is again, what target do you want to look at first? Right? GPCRs. Oh, that's a nice question by Faradilla. That's right. So there is a bias with the GPCR on the, for example, beta arrestin versus the side of AMP. It depends. So with the GPCR, for example, in our cannabimemetics drug discovery, we know that although we have two compounds that has very similar cannabinoid activity, but one, as you said, show more bias towards beta arrestin, for example. So I didn't go through that obviously in the beginning, but this is why when we are looking at GPCR modulators, there often needs to be more follow-up pharmacological assays, right? So emotional blunting, delayed onset, yeah, exactly. So especially for those GPCR modulators for CNS indications, we've seen that with the cannabinoid as well. Great question, Pak Radila. Isaac, oral ganti posisi apakah mempengaruhi efek apa aja dalam obat? Mostly the binding. If you're talking orthometapara, that's mostly the binding.
Dokter.
And Marom, what's the significance of functional groups? Why might some, in order to maximize binding, afra atayala marom, you change the functional groups so that your drug binds better to your target. Heavier organic molecules from most medicine. I think I answered that. Heavier allele in accordance to drug substance that doesn't fit. Can we still alternate? Yes. Allele, I think I've already answered your question. Similar question was asked previously. Barrel. When you're designing ACE inhibitors, you're adding that hydrophobic pocket. How do you balance this with the potential risk of reduced selectivity or increased toxicity? Again, quite typical. When we do one change, we do need to repeat the rest of the assays barrel. So at least in this case, we know that we get improvement in the ACE. So, but as you said, we do need to repeat the study in another panel of related receptors. Nadia Rahmania, how can we?
Dr. Hendra, we are very kami sangat bergembira ya. Ya banyak sekali pertanyaan dari mahasiswa tidak berhenti begitu. Akan tetapi sayang sekali waktunya sudah habis. Dan kami sangat menghargai pertanyaan dari mahasiswa ya. Kalau ada